Hello, my name is Anna Madisich, and today I will be covering the paper Flavonoid-attracted Aeromonas species from Arabidopsis root microbiome enhance plant dehydration resistance. This paper focuses on the plant microbiome niche. In essence, this paper underscores the pivotal role of flavonoids in facilitating interactions between plants and microbiomes, particularly evident in the enhanced resistance of plants to dehydration through bacterial involvement. Why does this matter? Well, the escalating impact of global warming is precipitating agricultural challenges worldwide, prominently including drought-related issues. To sustainably meet global food demands amid such adversities, crops must evolve to withstand these harsh conditions. While various strategies are currently in play to bolster crop resilience, exploring avenues like leveraging flavonoids for enhanced dehydration resistance presents a promising frontier. The insights gleaned from this research offer a broader perspective on plant-bacteria interactions beyond conventional nodulation pathways. They encourage us to delve deeper into identifying bacteria that specifically fortifies plants against dehydration, unraveling additional stimuli that plants exploit for synergetic drought tolerance, and translating these discoveries into agricultural practices. By leveraging this newfound knowledge to enhance soil quality and cultivate crops with heightened drought resilience, we can take meaningful strides towards ensuring food security in the face of climate challenges. This paper focuses on Arabidopsis thaliana, a renowned model organism in plant research due to its rapid growth and minimal space requirements. Unlike some plants, Arabidopsis does not form nodules during bacteria interactions, a key aspect that researchers aim to explore. An illustration of Arabidopsis can be seen on the left. The primary compound under investigation in this study belongs to the category of flavonoids, metabolic substances produced by plants in response to various stressors such as herbivory and drought. When plants perceive threats, they release flavonoids as a sort of distress signal to attract assistance. For instance, when plants detect herbivore activity, it releases jasmonic acid, known to recruit predatory insects like wasps to combat the herbivores. Similarly, plants use this mechanism to solicit help from beneficial bacteria. Zooming in on roots, one of their vital roles is to excrete substances into the soil. Among these substances, flavonoids play a prominent role, likely serving to attract bacteria and enrich the root microbiome, ultimately promoting overall plant health. Some plants take these interactions to the next level by forming specialized structures called nodules, which act as a cozy habitat for the bacteria. These nodules provide a concentrated space where the plant can benefit from the bacteria, while the bacteria find stable residence within the plant. This phenomenon is particularly common in the legume family, exemplified by nitrogen-fixing bacteria found in certain legumes, a direct outcome of the nodulation process. Of particular interest in this study is Aeromonidaceae, a gram-negative facultative anaerobe. Within this family, we'll specifically focus on Aeromonas species H1, later referred to simply as H1, which is readily cultivatable in laboratory settings. These bacteria exhibit intriguing behaviors in response to flavonoids, showcasing chemotaxis or directed movement. When exposed to flavonoids, there are a notable increase in transcriptional activities related to flagellar biosynthesis, enabling the bacteria to navigate towards the roots more efficiently. Despite this background information, some questions still remain. How do flavonoids affect non-modulating bacteria? Why does this cry for help strategy work in abiotic stressors? What are some of the main players in the root microbiome for helping with dehydration response? How do flavonoid-mediated plant microbiome interactions improve resistance to dehydration? These questions are some of what researchers are hoping to address within the paper. The researchers hypothesized that by augmenting flavonoid production, the root microbiome could be altered, thereby attracting beneficial bacteria and ultimately enhancing the plant's resistance to dehydration stress. In order to test this hypothesis, the researchers followed the following methodology. First, looking at bacterial strains, newly isolated Aeromonas species H1 alongside B. amyloliquefin GB03, B. Megatherium YCR R4, and B. Megatherium TGI1E were used. Strains were stored in negative 80 degrees Celsius and streaked for Luria bentani LB plates for regular use. For inoculum preparation, the bacteria strains were cultured in liquid LB medium until reaching exponential growth phase. Then they were centrifuged and resuspended in 0.45% NaCl saline solution for soil inoculation. For the plant material and growth conditions, Arabidopsis mutant PAP1D and TT44, coriander, and japonica rice were used. The seeds were sterilized and planted in half-strength Murashig and Shuk medium in petri dishes. Growth chamber conditions were set at 22 degrees, 16 hours a day, and then 16-hour night cycles, and 100 micromoles of 2 to the negative 1 s to the negative 1 light intensity. For the soil substrate, natural soils from Chenshao Botanical Gardens mixed with commercial soil to a 1-to-1 ratio was used to avoid stickiness. Soil homogenized and distributed to the pots with 4 biological replicates per plant genotype. For the microbial sampling preparation, DNA was extracted from the soil, rhizosphere, and endosphere compartments. The DNA was sequenced performing 16S rRNA gene metagenomic sequencing. A chemotaxis assay was completed by using natrigen, glucose, quercetin, camparifold to examine the potential chemotaxic attractions. Assays conducted using the Boyden chamber tested with 100 micromolar chemical solution. The biofilm formation measurements were taken by having bacteria cultured in the liquid LB medium with or without the compounds. Then the biofilm structure was stained with crystal violet and measured at 530 nanometers. For the RNA sequencing and DNA analysis, first the RNA was extracted from bacteria and Arabidopsis for the sequencing and analysis. Differential expression genes, DEGs, identified using the EDGE-R. Quantitative real-time PCR was completed, looking at complementary DNA synthesis from RNA samples and used for the real-time PCR. The gene expression was analyzed using the 2-delta CT method. The plant's beneficial traits were measured, including bacteria-produced auxin, siderophores, phosphate solubilization, ACC diaminazide activity, H2O2 catalase activity, and polyamine production. Bacteria enhancement of plant dehydration resistance was looked at by taking soil-grown Arabidopsis, rice, and coriander plants, inoculating them with bacteria, and subjecting them to dehydration stress. The bacterial root colonization was measured by looking at the quantification of bacterial colonization in the roots using CFU per milligram fresh weight. And then other steps were taken in order to look at different levels of success within photosynthesis. Diving into some of the key results, we see that in this figure, there is abundance of different bacterial families that was found within the 2 Arabidopsis types. In this study, Arabidopsis PAP1D mutant was characterized by its significant overaccumulation of flavonoids, and the col O represents the wild-type Arabidopsis with less flavonoids. In this graph, each dot and triangle represents a different family of bacteria. The higher up the dot, the higher accumulation was found in the PAP1D mutant. Among all the bacterial families studied, the Aeromonas exhibited a higher relative abundance in PAP1D compared to col O in both the rhizosphere and the endosphere. These findings suggest that flavonoids may have a preference for attracting Aeromonas within the root microbiome under the test conditions. This figure illustrates that Aeromonas species H1 exhibits greater colonization on PAP1D compared to wild type. Additionally, the flavonone naringenin triggered chemotaxin in H1, resulting in increased bacterial mobility. This is seen with the higher bar for the PAP1D. Similarly, this chemotaxic assay shows that H1 was attracted by naringenin more than the other flavonoids present. This can be seen in the cluster of black dots having the highest relative attraction. Overall, Figure 2 shows how H1 reacts to naringenin with increased mobility, causing increased colonization of Arabidopsis. Figure 3 now evaluates the dehydration resistance seen in plants with H1 colonization. Figure A shows pictures taken at different points of exposing Arabidopsis to drought conditions. On day 0 at the top, we see how the entire row looks relatively uniform. However, by day 14, shown in the middle, we can see a notable difference between the control and the H1 group. The control on the left looks much more dried and impacted, while on the right, the Arabidopsis inoculated with H1 show much greener and hydrated looking leaves. This shows how H1 increases resistance to dehydration stress. Figure B assesses the photosynthesis efficiency in both groups during the drought treatment. Arabidopsis plants inoculated with H1 exhibited less reduction in photosynthesis efficiency compared to the controlled plants. Soil inoculation of H1 significantly enhanced the dehydration resistance in Arabidopsis, as evident by the marked improved plant survival and photosynthesis efficiency. This is seen in the blue H1 line remaining higher than the green control line towards the end of the drought treatment. Figure D presents the combined outcome of H1 presence or absence alongside different Arabidopsis mutants. The figure examines the survival count of plants per pot under drought conditions with these varied treatments. The Arabidopsis TT4 mutant lacks flavonoid production compatibility. H1 colonization failed in the dehydration stress TT4 mutant, resulting in no bacterial enhanced dehydration resistance. Thus, the flavonoids are essential for strain H1 enhancement of plant dehydration resistance. Overall, Figure 3 suggests that beyond increasing flavonoid accumulation in plants, dehydration stress induces certain other effects that counteract flavonoid-mediated attraction of H1. Nevertheless, flavonoids remain crucial. Finally, looking at Figure 4, we see an RNA-seq analysis of Arabidopsis transcriptome showed how plants respond to dehydration stress in Aeromonas species H1. The Venn diagram at the top indicates the number of genes involved, which were further analyzed using K-mean clustering and gene ontology, GEO, analysis. This analysis highlighted a group of genes related to jasmonic acid, JA, known for promoting plant resistance to dehydration stress. These genes are enriched in functions related to stress response, secondary metabolism, defense, and signaling processes, as revealed by geoanalysis. This helps to illustrate the synergetic effect that many factors play in helping with drought resistance. The study demonstrates the crucial role that flavonoids have in mediating plant microbiome interactions, particularly in enhancing plant resistance to dehydration stress. Flavonoids attract beneficial bacteria, such as H1, which colonize the root and contribute to improved plant resilience. H1, attracted by flavonoids, displays increased colonization on plants with elevated flavonoid levels. The bacteria show enhanced mobility in response to flavonoids, indicating a chemotactic response. The presence of H1 increased plants' dehydration resistance, as evident by improved plant survival and photosynthetic efficiency. However, the enhancement was dependent on flavonoids, as strains unable to produce flavonoids showed no improvement in dehydration resistance. RNA-seq analysis revealed synergetic response of Arabidopsis to dehydration stress and H1 colonization. Genes related to jasmonic acid metabolism and signaling were upregulated, indicating their role in promoting plant resistance to dehydration stress. DEGs associated with stress response, secondary metabolism, defense, and signaling processes were enriched, further highlighting the plant's adaptive response to dehydration stress and bacterial colonization. Overall, the study underscores the importance of flavonoids in shaping the root microbiome and enhancing plant resilience to dehydration stress, while also elucidating the molecular mechanisms underlying these interactions. While the research presents valuable insight, it's important to note some limitations. Firstly, the study focuses on specific plant species, so the generalizability of the findings to other plants needs further exploration. Additionally, the mechanism underlying the reduction in strain H1 colonization under stress conditions remains unclear, highlighting the need for further investigation. Finally, the study mainly discusses the interaction between flavonoids and bacteria in enhancing plant dehydration resistance, potentially overlooking other factors contributing to this process. These limitations indicate areas for future research and refinement of the current findings. Some future areas of study could include exploration of other plant microbiome interactions beyond flavonoid-mediated ones, investigation into specific mechanisms underlying the reduction in strain H1 colonization under stress conditions, examination of the broader implications of plant bacteria associations in various environmental stress responses, development of strategies to harness plant microbiome interactions for agricultural and environmental applications, and integration of omic approaches to gain a comprehensive understanding of the complex crosstalk between plants and their associated microbiomes. To reiterate, these results can hold importance in increasing our understanding of flavonoids in the microbiome, which can be applied to making better soil for crops in order to help their survival in the face of global warming. Furthermore, there are many reasons we should care about these results. Understanding how specific bacteria like H1 can improve plant resistance to dehydration stress and offers potential agricultural benefits by enhancing crop resilience in water-limited environments. This study sheds light on the intricate interactions between plants and their associated microbiomes, particularly regarding the role of flavonoids in shaping microbial communities and promoting beneficial associations. Discovering the role of flavonoids in attracting beneficial bacteria underscores the ecological significance of plant metabolites in shaping microbial diversity and function in soil ecosystems. Insight gained from this research could inform the development of novel biotechnological approaches for crop improvement, such as the targeted manipulation of plant metabolites to enhance beneficial microbial associations and improve stress tolerance. By elucidating mechanisms by which beneficial bacteria enhance plant resilience to environmental stressors, this study contributes to the development of sustainable agricultural practices that reduce reliance on chemical inputs and promote ecosystem health. And lastly, this research advances our fundamental understanding of plant microbe interactions and stress response, providing valuable insight into the complex networks that govern plant health and resilience in changing environmental conditions. and here are my references.